Welcome back for week 2 of digital learning. Today, our focus is going to be on the reading notes that you completed on Friday in terms of some of the vocabulary that you should be familiar with. This also is going to be a little bit of a review from other assignments you completed this past week. I also will then go over different sample problems within this for you to hopefully better understand these principles that we'll be talking about. And what I would like for you to do on pages 66 and 67 of your bill is I want you to add these notes as I go through them with you on this presentation. So please use page 66 and 67 of your bill to take the following notes. So the first thing, again, basic vocabulary. So you should be familiar with these terms, but if not, hopefully this clears it up. If we're thinking about heterozygous has other terms that mean the same thing. So heterozygous is where you have two other or different alleles, and that is going to mean the same thing as a hybrid. Now, hybrid specifically refers to the organism, but the organism that is a hybrid will have a heterozygous genotype. Therefore, they can be used interchangeably when you're doing these Punnett square problems. If I use the example here between these two plants and I'm looking at the gene that codes for height, that there can be a gene variant or allele that's dominant and an allele then that would be recessive for the short height. An example genotype that would be heterozygous would be big T, little t, so two different alleles. Homozygous has an alternative meaning. Again, we use this term when we refer to the whole organism, but again, this organism, it would have a homozygous genotype, so they could be used interchangeably. The vocabulary term here is pure. So if an organism is pure, it has a homozygous genotype. The individual, in this case the plant, could have a homozygous dominant genotype. It's going to be pure for that dominant allele, or it could have a homozygous recessive genotype, in which case then it only has two little letters, so it only has that recessive genotype. So here's a sample problem. How many offspring will be short if a pure tall plant is crossed with a short plant? So just like before, we take one parent. Let's do the pure tall, I think is the one that I have first. And the pure tall would be homozygous, because they mean the same thing, for the tall allele or the dominant allele. And I put one allele over top of this box and one allele over top of this box. And if they're crossed with a short plant, well, the only way that an individual can show the recessive phenotype, being short, is if they only have recessive alleles. So here we have two recessive alleles. And again, I put one T here and one small t here. So now we need to fill it in. So to fill in this first box here, it's going to be big T, little t in this box, big T, little t in this box, and big T, little t in this box. So then again, if I'm using my rules, I answer the question. The question is how many will be short? Well, if I'm looking at each of these boxes, again, the only way that it could be short is if it has two recessive alleles. And I don't see any of these boxes that have two little t's in them. So therefore, 0 out of 4 would be short. So therefore, 0% of the offspring would be short. Incomplete dominance. Incomplete dominance, if we were to just kind of briefly summarize it, I might say that it's an inheritance pattern where both alleles are expressed as a mixture. So here's an example. Let's say I have this family here. And if I'm looking, I might have a T allele for tall, an S allele for short, but then if the individual has one of each, they kind of have that mixture or a blend between each of those alleles that they inherited. So 2 more words, just a quick review. If I'm looking at the physical traits of the organism, remember that that is called the phenotype. And if I'm looking for the genes or alleles that make up a trait, that is called the genotype. So sample question. If 2 medium height parents mate, what percentage of offspring would also be medium? Well, if they're medium, I see it right here, it's going to be that mix, so therefore they have to have one of each of the alleles. So I'm going to put one of those short parents at the top. I'm going to put the second short parent off to the side. Then I'm going to fill in my box. So SS, S or TS, ST, and TT. Now, one thing just to note is I do have the S and the T in the same order instead of switching them around. And just for my sake, it helps me recognize that they are the same. So I would recommend doing that when you solve your problems. Keep it as a consistent format. All right, going back and looking at the question, it says, what percentage of the offspring would be medium? Well, medium, again, I know the genotype is ST. Looking at the box, I see 1, 2 of the 4 that are ST or medium. Therefore, 2 out of 4 will be ST or medium, so that the answer would be 50%. Next is codominance. Codominance is not true Mendelian because they don't have one allele that would completely mask or hide the other. So if we were to quickly define what codominance is, I would say that codominance is when both alleles are equally expressed. Now, the example here that we have is blood type. So blood type is codominant because if I see here, if I have an A allele, then I have a particular A receptor on my red blood cell. If I have a B allele, I have a B receptor on my blood cell. But if I have both A and B alleles, I don't have a mixture or a blend or just one or the other, but I have both. So both the B and the A alleles are equally expressed on the blood cell. There is also a recessive allele in this case, and that is O. So if I look at O, this one doesn't have any of those receptors. So in this case, O is considered recessive because the only way that you could have an O phenotype is if you only have O recessive alleles. So because we have A, B, and O, this is also considered a multiple allele trait because there are more than 2 alleles for a trait in a population. Even though each individual will only inherit one of those alleles from each of their parents, so they only have 2 total, in a population you can find more than 2. Okay, so the first question here, genotypes. What are the genotypes of these? So if you have type A blood, there are 2 possible genotypes that you could have. You could be homozygous or pure for A, so you could have big A, but you could also be heterozygous because there is a recessive allele in this combination. So you could be AO. And the way I like to do blood typing, I have A and B, I'll write as an uppercase letter, but my O I write small to show that it's dominant, or to show that O is recessive. All right. For type AB blood, what are my genotypes? Well, there's only one. It has to be AB. You have to have an A allele and a B allele to be type AB blood. For type B, this one you can also be a heterozygous for. So you could either have BB or BO alleles that would make up a type B blood. And O, only one combination. You have to only have those O recessive alleles. Again, because the only way that you can show a recessive phenotype is if you only have recessive alleles that make up your genotype. Okay, so sample question. If one parent is heterozygous for type A blood and the other is heterozygous for type B blood, can they have a child with type O blood? So figure out genotypes of parents. It says heterozygous for A, so I know that it has to be this one. So I'm going to put that one at the top. Heterozygous for B, again, I know it has to be this one. It cannot be homozygous. So I'm going to put the BO on the side. And then I'm going to solve my problem or fill in my square. So that would be AB, BO, AO, and OO. And again, go back to the question, can they have a child with type O blood? Type O, 2 O's. So the answer then would be yes. There is a 1/4 or 25% chance that they could. And then the last type, I'm going to start with this question. So can you read these numbers? I'll give you a minute here. So maybe you can see these, maybe you can't. This is 12, this is 2, this is 42, this is 74, and this is 6. So this is an example of a sex-linked trait. So red-green color blindness is an example of this. So to put in your bills, what is a sex-linked trait? A sex-linked trait are traits that can be found on either the X or the Y chromosome. So those are your sex chromosomes. Therefore, if they're attached, they are linked or they are found there. One thing to note when you're doing sex-linked traits is because they are found on the X or the Y chromosome, you have to keep it attached, because if I'm looking at males versus females, especially if it's on the X chromosome, females have 2 of them, so they would have 2 alleles, whereas males, if it's found on the X chromosome, only have 1 version. And then if it's found on the Y chromosome, well, only males would be able to have that allele. Females do not have a Y chromosome and therefore would not have those alleles. So the question, if the male is colorblind and the female is a carrier, what percentage of their children would be colorblind? Before I even go to the rest of the questions, let's figure out the genotypes. So male colorblind. In this case, if you are colorblind, that is a recessive trait. So the male would have a recessive allele attached to his X chromosome. I probably should have also mentioned that it's an X-linked recessive, so it's not going to be found on the Y, it's found on the X. So when you write the genotype here, I write the X, and as a superscript, I write that little allele, so the lowercase b allele. Because he is colorblind, he has to have that recessive allele. And the Y doesn't have anything attached to it because this allele is not found on the Y chromosome. For the female, it says she's a carrier. So here's a term. Carrier also means the same thing as heterozygous or hybrid. Carrier means that they have 1 dominant allele, but that they carry or hide that they actually do carry the recessive allele. So the female, because she's got 2 X's, I put a capital B on one of her X's because she's not colorblind, and I put a lowercase b on her other X because she's a carrier. So she does have one of those recessive alleles. Even though she is not colorblind, because of this dominant allele, she does carry that allele for colorblindness. Okay, so I'm going to put them on the chart. I'm going to put the male here. So again, separating his X and Y chromosome, and the female at the top. Then I'm going to fill in the Punnett square. And now I'm going to go back and answer the question. So there's 3 total here. We're going to start with the first one. So the first one says, what percentage of their children will be colorblind? So to be colorblind, since it's a recessive trait, you have to only have recessive alleles. This one has a dominant allele, so it will not be colorblind. This one only has recessive alleles, so this female would be colorblind. This male only has a recessive allele, he would be colorblind. This male does not have a recessive allele, therefore is not colorblind. So total, there are 2 out of 4 of their kids, and that would equal 50%. So the second question then said the percentage of girls that will be colorblind. So now I'm only going to look at the top 2. Only look at the top 2. So if I'm looking for a percentage of females, I'm not going to look at the males. Of the females, 1 of 2 are going to be colorblind. Therefore, 50% of the girls will be colorblind. And then the last question, what percentage of boys will be colorblind? So again, I'm only going to look at the boys, and I see 1 out of 2 will be colorblind. Therefore, 50% of the boys will be colorblind. Okay, so hopefully, again, you put these notes into your bill on pages 66 and 67. In Schoology, I have several assignments then for you to complete this week. One is a ed puzzle on incomplete and codominance. Another one is a non-Mendelian vocabulary quiz. And then the last one is some practice problems. So they'll be for codominance and incomplete dominance. If you have any questions, please let me know. Have a good day.